So today, like our specific focus is on our recent work that we have been doing. It's called AgForce, short for antigen forcing mechanism that that addresses some of the failure modes in current antibody design methods. And here we use some of the concepts from multiple choice learning and mixture density networks to develop some specialized architecture. So yeah, let's get started into like first let's like for some background what are antibodies and like why are we like interested in them basically. So you see here we have all seen this picture. Maybe we have bad memories from the COVID pandemic, but we have all seen this SARS-CoV-2 and the red circular thing inside, and we see the white things that are attached to it. Like so those are called basically like so antigen so SARS-CoV-2 is basically an antigen or a pathogen that enters our body, and then our body naturally produces antibodies to attack those foreign substances and then neutralize them. So sometimes our bodies are not able to produce those antibodies, which makes us sick. So for example, like we have cancer or we have like all kinds of HIV or like SARS-CoV-2, like, and then they make us sick. And then, like, for some definition purposes, like, these pathogens, these foreign substances are called antigens that we will refer to in the entire discussion. And then antibodies are what our bodies produce, our immune system produces. So those are like our targets. Those are our target biomolecules that we're interested in to be developed and designed later on. So currently, like as we all know, like in like for centuries, for like decades, antibodies are produced in wet lab and where animals are immunized for whenever certain antigen comes in. So these animals are immunized, and then after a few months or a few weeks, those antibodies are taken out and then they're processed in the lab. So it's like mostly happening in vivo, within these animals. But the caveat is that it's like it costs like billions of dollars, and it is a very time-taking process, and and like sometimes we can't afford, for example, in the case of COVID pandemic, or if we have like a lot of like other diseases that we still don't have cure to. So we think that so before like moving into the computational aspects of it, let's set up some definitions for like what an antibody, how an antibody looks like, and so that we are more focused on the type of data that we want to work with. So antigen is like antibody is basically this Y-shaped thing that you see here. It has like two chains, like two chains of amino acids, heavy chain and light chain. And then there's an antigen that binds to it on its tip. So you see the red color thing is the antigen that binds to this specific locations in the antibody. So if we zoom in, so this region is only important to us where the antigen is going to bind to this antibody, and this is what differentiates like one antibody from another antibody. And so all those properties that are inherent to that particular antibody, like for example their binding affinity, or like how strong its binding affinity is going to be, what is how its sequence is going to look like, how its structure is going to look like. So we there's a special term for it. It's called complementarity determining regions. So CDR regions are the regions, they are like subsequences within the antibody tips. Like they lie on the tips of the antibody, and they are called CDR regions. They have like we have like small substrings called H1, H2, and H3 because they encode into different genes. They are encoded through through a separate genes produce them. So they are like crucial to understand whenever we are talking about antibody or antibody design in general. So like just for the setup here, because these are like proteins, so we have its 3D structure, three-dimensional structure, PDB, and then we have its sequence. So we work with like two different modalities or two different representations for the antibody, and this is what we're going to do, and it's a crucial aspect within antibody design. So like how are so like as I was talking about that there is like we are only interested in like in the tip regions like wherever the antigen is going to bind to antibody. So they have like a lot of known properties that we already know about. So because an amino acid is basically like a protein is made of like amino acids that are bound together with this peptide bond. So there's like a chain of amino acids that stitched together and then they fold into some specific shape. But when they bind to the antigen, so there is certain properties, for example, hydrogen bonds that take place between the hydrogen atoms of the antigen and antibody. There is van der Waals forces, there's hydrophobic, electrostatic interaction. So there's different types of those interactions, and we have some special name for the binding site, the binding location in on the antigen, which we call epitope, and then on the antibody side, it's called paratope. So just for the sake of definition, it's a patch of those amino acids that cluster together, and then they are like the target location for this lock and key mechanism. Whenever the antibody is going to bind to this antigen, so these patches of amino acid, they are important to us. So if you zoom in more, and we will see that. So here's like how like an antibody an antigen interaction looks like. If you zoom in, there's all sorts of different bonds, and those are really crucial to understand before we develop some more before we model them basically. So now that we have like the background and setup for like what we are looking for in antibody design. So here we see that there is opportunities in terms of computational modeling here. Because we can define something, we have some data, we can like write the math for it, and then we can do some kind of training, do some prediction or designing on top of it. So the input here is going to be like whatever antigen, the target antigen that you have. And you don't know its antibodies that are going to bind to it. So those are unknown to us. So that you see like these question marks here, and then your model is supposed to, your software is supposed to predict what do those antibodies would be that are going to bind to it. So like for each specific like I mean like epitope is like the binding site of antigen. So you specify what its epitope is going to be, where do you want the antibody for, because you have to specify that an antigen can have multiple regions where different antibodies are going to bind to it. So their sequence and structure are going to look different. So here, like you see, like you're also given some part of the antibody. Like for example, if you remember from that Y-shaped, the rest are like there's a fixed region in antibody that doesn't change from one antibody to other. Only like the CDR regions are going to change. So that's our focus, and that's our point of interest. We don't, so we give that as input to the model as well. So if we define the problem like more like informally, we are given the antigen, so antigen structure and sequence basically, and then we are given the epitope, and then we are given the constant region, the framework region of the antibody. So we are given three things as input, and then our output is supposed to be like what do those like fill those CDR regions basically, like both its sequence and its structure. So that's like the end goal here. Antibody design has been like around for a while, like for the past 2 3 years there has been significant progress. So there are like different types of like paradigms of modeling or generative modeling have been proposed to do like to work with or to develop better and more efficient systems that can do the antibody design for us. So one of them is like the classic like graph neural network based approaches. So because like natural representation for proteins is like, as you might know, is a graph basically, because you have amino acids and then like a residue level graph, you have amino acids and then you have edges between them. So like nodes and edges, they're like a very natural representation to represent a protein. So because antibodies and antigen are also proteins, so what you do is you do graph reconstruction here. So graph reconstruction is like you don't know like what do though like in the antibody like what do like certain nodes would be. So like you like initialize them randomly and then after certain refinement you do like reconstruction there, and then you predict with some objective function that like what's it gonna be like. So like you can work with all different kinds of GNNs here. The other like represent the other modeling paradigm that has been like around for antibody design is through diffusion models or these sampling based approaches, basically where you learn the data distribution in general, and then you sample like candidates from it and then rank them based on some like energy function, some objective that you want to optimize for. So sampling based approaches have also been around, like diffusion models, flow matching, and there's like different variants of it. People also do like retrieval based diffusion or like yeah, like so the third way is diffusion is basically like the denoising based where you denoise and then learn the data distribution in general. And then like the third one is basically like autoregressive based approaches where you like predict the next token, and usually like you work with some pre-trained 14 language model and then fine tune it on antibody design and on antibody sequences. And so these are mostly like they're only going to give you the sequence, not the structure. So what we did was what we want is we want a co-design approach. We also want the sequence and we also want the structure, the corresponding structure to it. Because if we have the sequence, we also need the structure. So the structure is really crucial to understand the binding here or like interactions here. So because like so these different types of modeling paradigms have been around, what we realized is we did some benchmarking and some failure mode analysis of like different types of these methods that have been proposed in the past 2 3 years. And what we did was like although like antibody design has been around, it has significantly been bottlenecked by the limited amount of data. So think of it like it's like the maximum data that we currently have that we recently benchmarked is 3,000, around 3,000 examples only, 3,000 antigen antibody complexes. And the job is to learn or to train some model only on 3,000 examples. You might understand that for a machine learning model, it's like it's very very minimal like to learn to learn some good function here that can do the design job for you. So that's our constraint here, and so we have to work in within this constraint. That's our like data set availability. So what we did was like in our like antigen force paper, we did failure mode analysis of like different antibody design methods, the GNN methods, the graph methods, the sequence-based, the diffusion methods, and the graph-based methods. And what we realized is that there is like a failure mode in terms of the sequence that they are going to output. So like we identified three main failure modes here. Like one of them is these models, because like even though like they work with specialized architectures, like they forget the input antigen, whatever input antigen they're getting. So essentially think of it like for any antigen, these models usually give you the same antibody. That doesn't make biological sense, because each antigen should have, and its epitope should have a unique antibody for it, and there should be diversity in your outputs that you're producing. So we realized that when we swap like with some random antigen, usually the output is was the same. So that's a huge failure mode because they don't make biological sense. And the second is like what we call like the vocabulary collapse mechanism. So we formulate the math for it that this is basically like because that because there's no diversity. So let me show you a picture of what do I mean by here. So what you see here is these are like heat maps, like the frequency distributions of the sequence of CDR, whatever we want to output. And you see that so on the x-axis, these are like the positions. Like there's like a defined like set of positions. These are very small substrings, by the way, like a 20 amino acid substrings, CDR H3, H1, H2. So they're like small substrings. What when you like take the ground truth data, your actual data distribution, and then you plot its frequency distribution at each position. So on the y-axis is like the 20 amino acids that we have, and on the x-axis is the position, is their position in the sequence. So in ground truth, you see that there is enough diversity, and most like at each position, like amino acids are like there is different amino acids that are occurring in the ground truth data. Like some of them are more frequent, some of them are less frequent, but they still exist in the data. It's not that every time. So this is like the ground truth, and then like all the like everything you see like after the ground truth is like it's some baseline method that we benchmarked, and the last one is our method, Antigen-force method. So there is a trade-off. So you see that these the red colored like methods, for example, rod, mean, dimean, or ABOD, these are different methods. These are mostly like graph-based methods, and then the green ones are flow or diffusion based methods. You see that or like autoregressive methods. So we observed a trade-off here. The methods that are accurate, like they make like some accurate predictions. For example, they have like higher recovery rate, sequence recovery rate. They collapse to some most frequent amino acid. They just produce that all the time. For example, rod produces Y like 80% of the time, like just like Y's. And but it has the highest recovery rate. Mean similarly like these methods. And on the opposite, you see the ones like in this scalar plot, the ones which are on the right side, so these are like the flow or diffusion based methods. They have a lot of diversity, but they're not correct. The predictions are not correct. So we see that there's like a trade-off, like either you're accurate and you're just producing like same output, or yeah, so and either you're like a flow-based method and you're producing diverse outputs, but they're not correct. So we want something that's in the middle, that is diverse as well, and that's also like accurate as well. So that's what like we wanted to focus on here. So I'll go to like our like we formulate it like mathematically and give like propose some like theorems to like that why this happens, and we identify that this is because of the cross entropy law. So like you might all be familiar with cross entropy is basically like the objective when you have like categorical outputs that you want, and cross entropy is basically like it matches the distributions, and that's the objective that you want to minimize. Like it's binary cross entropy loss. Like binary is like when you have two classes only, but in this case we have 20. So like our cross entropy is like for 20 classes of amino acids. Um, what we realized is that because these methods, they're still getting the constant framework region as input. So, like, if you remember from the Y-shaped antibody, we want to design the tips, and the amino acids that are on the tips of the antibody. But we are also provided the rest of the framework as context. Because that framework that we're providing it is constant, so these baseline, these methods, they usually like bypass the antigen, and they are expecting every time there is something constant in their input. But that's also important to model as well. Like the model needs to understand the entire the chain context, because it's taking like the context as input, that's constant all the time. So these models with cross entropy loss, with simple cross entropy, they just resort to the global maximum of cross entropy, and then that's the argmax of each position basically. So whatever is the most frequent, they keep on predicting that, and they don't even like look at the antigen because this is like a bypass path here. So this is like the broad intuition for it and what we like formulate theoretically, and so we try to like improve these and like to cater to these like failure modes in general, and also keeping like a good accuracy that we want, like so we want like something that's in the middle, and AgForce is like we are able to do some improvements here. Um, so let's look at like the
Mansoor,
any questions here? Yeah.
Mansoor, yes, on the previous slide. So the models that of the graph and the diffusion based models, I think I have missed this, but I would love to know which ones are predicting like the most frequent. And I feel like if the performance is like this, I would think that this is poor performance, like they're like predicting within the distribution almost that it was used in training. So I would want to know of the graph type and the diffusion based models, which ones are frequently doing this of predicting like the most frequent um, sequences or structures.
Yeah, that's a great question. So, the graph-based methods, they usually have this behavior. They predict the most frequent ones. And the diffusion-based or the sampling-based methods, they have a lot of diversity, but they're not accurate. So usually, like because the diffusion-based methods, the sampling methods, you have to like learn the data distribution, and for this amount of data, this is not the failure of the inherent failure of these models in general. This is the failure of the setup that we have. Because we have like, very less amount of data, we have to tweak these architectures. You can't just like implement them off the shelf and like hope that they will work. So like the graph-based methods, they like even though like in other domains, when you have a lot of data, they can learn well and they can do like do a good job. But in this for this particular task, you have to understand that like these kind of failure modes that you're encountering. So they make like the modeling job difficult. So the graph-based methods predict the most frequent ones, and they have like very low like what we call effective vocabulary. So like vocabulary is like for example in language modeling, they just like predict the most frequent ones all the time, and there's like no diversity in their outputs. You see like like for example like the three methods on the top here, like rod, mean, and dimean, they're graph-based methods, and they like you see like, it's all empty like in all other for all other amino acids. They just predict like the most frequent ones and all the time. So that makes like their outputs like not very meaningful, but they still manage to have good recovery rate. So this is also a failure mode of the like metrics that we evaluate these methods upon. Even though like their outputs don't make sense, but with our definitions of, because we do like with recovery rate, we just compute like match the like the ground truth and the predictions, and then we compute like how many of them are correct. So usually like the frequent ones, because they're the most frequent ones, they're this is just like a random guess, and they happen to have good recovery rate. So this is like also like a failure mode of the like recovery metrics. So you also have to look, and I'll talk about it like later on, is that there's no single correct answer here, like for each position. So like there can be multiple correct amino acids that your model should be able to predict, and they would make sense because all that matters is the binding here. Like so with binding, for example, leucine and cytoleucine, they can both be should be like likely. Like But instead of like leucine, for example, there is some other, let's say hydrophobic interacting with another hydrophobic amino acid in the antigen. Those should be very less likely. So that's what like I want to like model. I want to talk about like how we approach this problem and how we try to like solve it. So yeah, that's any other questions?
I think no.
Yeah. So yeah, let's jump to the architecture, and then we will talk more about the results as well, like what we were able to do there. So again, like this is like the classic setup that we already mentioned. You are given the antibody, the blue-shaped white thing, and then on the tips, the question mark region, those are the CDR regions that are unknown, and you're given the antigen, and then you're given its binding location, the orange region. So what like as like computational scientists, we are like our like go-to approach is that do some representation learning, do some encoding, and then predict whatever you want as output in some decoder here. So like encoder and decoder architecture. So let's zoom in inside the encoder first. So with encoder, we didn't do much of the changes, but we did like try to model that bypass paths, and then we did like some tweaks here that we think like they work and they're able to like solve some of the failure modes. So with encoder, like the simple idea is that you have a graph neural network, like represent it as a GNN, and then apply some cross attention between the whatever your GNN is going to predict, like those like the blank like amino acids, whatever the GNN is going to predict, apply some cross attention between the GNN output and then your antigen, basically. So do some cross attention here, and then pass on these refined representations to the decoder here. So if you like zoom in here, this is called like a virtual node EGN. And so virtual node is basically like because we don't know, like, so it's like it's not a real node. It just, we want to model the interactions here, so we initialize them with some virtual node. But the catch here is that we know the length of CDR region. So they have like defined lengths. So that's like a good thing for us, because if it's like a variable length, then it would have been some trouble as well. So let's see like how like what we do here. So we do like before, like, so within our GNN, we do like we apply the idea of dropping out some like kind of removing some nodes during training, so that like it's like a regularization approach, so that the model is like more robust towards like not like memorizing the data, not memorizing the graph structure, but it should be able to reason, like if some of the nodes are missing from the input during training, it should be able to still understand the rest of the structure. So it's not like memorizing it. So this is like the classic, because it's a problem in GNN called like overscotching problem. So because like GNNs, like usually they like, so you want it to be like more generalizable. So that's the first idea that we implement. And this is like the definition for if you are familiar with like how GNNs work. So basically these are message passing networks. You have a node, and then you have the neighbors, whatever nodes have like edges between this particular node. So for example, take a node I, and then it has like neighbors J around it. So we like you aggregate their messages, basically like their embeddings together, and then for each node you do this process. So this way like and the embeddings get processed after certain layers. So after each layer, these embeddings get processed based on its neighboring nodes. So you initialize them with some like geometric features. For the amino acids, you can also use protein language model embeddings to initialize these nodes, basically, or these amino acids. So the third thing that we do in the encoder is what we notice is that people usually like use like a simple vanilla cross attention, where we all know we have like queries and keys. Um, what we did, what we noticed is that we also noticed some improvement here, and we have like a biological intuition for it, why it works like a bit better. So because these interactions between antigen and antibody, so like in vanilla cross attention, there's like we work in Euclidean space. So you you compute the cross product between the QD and Q, right? But they like the assumption is that the data is in Euclidean space. But we think that because these interactions between antigen and antibody, they can be like interpreted as being in a complex manifold. So in that manifold or in that complex geometric space, like why we think so? Because there can be multiple interactions that are possible. So there's not like one interaction only between an antigen and antibody. Like for example, it's like a hydrogen bond. It can at the same time be electrostatic interaction. It can at the same time be like van der Waals force. So because these interactions, they can be like multiple, and they can be like there's no like one-to-one interaction. There's like, so that's why we think that like a complex manifold, in this case, for example, Lorentz hyperbolide, it's like a sphere that we map the cuties and keys to, and then we do apply the cross attention in that space basically. So there's nothing different apart from the projection that we get. Rather than like the simple Euclidean cross attention that we do, we project the cues and keys to that Lorentz hyperbolide and then do the cross attention there. So it kind of captures that complex geometric or the complex interactions that can take place between the antigen and antibody. So that's something that like yeah, we tried, and it kind of works well. So now that we are done with the encoder,
Mansoor,
yeah.
Mansoor, yes, on that very slide, yes. I'm trying to like someone who naively coming from a computational end, but tries to understand what is happening at each layer. Like, so we have the antibody and the antigen, and I would love to get some light on the mapping directly into the EGNN. Yes,
Mhm.
we know what are the nodes in this case, what are the vertices to break that abstraction. But also, maybe something that you would add on is when you look at the hyperbolic attention, it is a function of Q and K. And I'm wondering, like, what in terms of the entities that we are looking at, the attention, the Q and K, what do they really represent in terms of the antigens and the antibody, and probably maybe the sites that we are looking at. So that's the kind of abstraction I would love you to break.
Yeah. Thank you for the question. Yeah. So, like, if we like look into the GNN architect, GNN itself, and what are the inputs here? So basically, like, uh, we have to understand what is given to us, right? So we are given the antigen. Antigen has let's say like m amino acids, and so we call them like m nodes. We construct m nodes, and then we define edges if they are like close to each other, if there's like peptide bond. If they're not, you can also do multi-relational edges here as well, like not just one single edge, but you can have multiple edges as well. So you construct the graph for the antigen. It's like straightforward, right? Like, and then for each node, you initialize some embeddings. Like, for example, what do serine have? Like, what kind of like, or in this geometry? So you compute like what should be their like surface area, what should be their like all like distances, and like all those kind of like geometric features that you can compute. So that will be some initialization for the antigen. You construct the graph for the antigen. For the antibody, you also initialize your antibody graph, but because you have some nodes that are missing, that are unknown, that's your whole point, right? Those CDR nodes that are unknown to us, you initialize them randomly, like with all like ones, and then for the rest of antibody, you also compute their geometric features. So you get like heterogeneous graph where there is like antigen, there's an antibody, and but you don't know their inter edges because like these are two different proteins, right? Antigen, antibody, two different proteins, two different graphs. But when they form a complex, when they pair together, they also produce it should have those inter edges between each other. So edges, like they should have bonds between each other, right? You don't know that. So in this case, what we do is we initialize it with some virtual node. Virtual node is like we don't know, so those are like the model will learn it. This representation for the virtual nodes. So
So they
and then
represent the
it will add.
epitope.
Yeah. We represent the entire antigen. We represent the entire epitope, in entire antibody. We create like this heterogeneous graph, and we pass this through the GNN. So GNN, basically what GNN does is for each layer of the GNN, it takes a node I. So what are we given, right? We have M nodes of antigen, we have N nodes of antibody, okay? And so there's a adjacency matrix, like the edges between them too as well. So it kind of becomes a bit like complicated, but to understand in like more simple terms is that GNN work on the principle of message passing. So message passing mechanism refers to like, so you see MIJ here. MIJ basically is like the message between node I and node J. And what it does is it aggregates the embeddings. Like you have some vector embedding for each amino acid, for each node. It aggregates that to the antibody, to the node I. So from node J, aggregate it, like add it to, you can do add, you can do averaging. There's different tricks to it. And you add those embeddings to the neighbor I, and then keep on doing for all the neighbors of node I. Like there's like, let's say like 5 neighbors. You aggregate them. So there's like an MLP, like that. Here's what the learning is happening, right? So MLP learns that how much weight should be given to a certain neighbor. So how much so this is how it learns its properties, basically, for example, this amino acid has this kind of neighbors, so they have these properties. And then after each layer, these embeddings get processed. So think about it like initially, like the model that didn't knew anything about its context, like because it got the raw input, and then after layer one, it attended to all its neighbors. It aggregated its messages. So now it knows its immediate neighbors in layer one. In layer two, same thing basically. So for all like neighbors of its first neighbors. So like it goes like it's like a K-hop neighborhood thing, like until the entire graph is captured. So this is how it learns the graph structure, basically, like the GNN itself. So it like for each layer by layer, and until like a few layers, let's say like 4 or 5 layers in our case. So it depends on how big your graphs are, like or your inputs are. If you have like millions of nodes, you should be doing like many layers. But if you have like thousands of nodes or like hundreds of nodes, few layers, and then you won't be overfitting. So after like a certain layers, you get processed or refined embeddings, basically. So like refined embeddings for your input. So you keep on doing it, like after certain steps, after certain layers, you get some output. Now, now that like you got these like processed embeddings, now do you can do simple cross attention, right? Because you have like M nodes and like M antigen nodes and antibody nodes. So take one of them as the query and the other one as the keys, right? As we do in cross attention, right? So you compute like so now you're like interacting like them each other. Previously they were being encoded, like just learning some representation. Now cross attention is going to learn like which like the interaction between them. Like is like going to learn that an hydrophobic residue on amino acid, on antibody, and hydrophilic on, let's say, antibody. So they should have a bond, they should have more attention score, the other one should have less attention score. So kind of it will learn the attention map or the similarity or the yeah, like, so like that's the job of the cross attention here. So query and key is were basically like in this case, like we take the antigen as the keys and antibody as the queries. It's your choice. You can do bi-directional attention too, and then dedicate them together. So you can do both as well. So yeah, so here the only difference is that we project before computing the attention scores, before like computing the attention, we project them to this Lorentz hyperboloid. So it's like a complex manifold. It's not like so. The assumption is that they are not in a Euclidean space, but they are in a complex geometric manifold. It's all because of these interactions. So that I earlier talked about, there could be like multiple interactions that are possible between two amino acids. So that's the idea. On a complex manifold, you can like capture that space. Yeah. Any other questions?
Yeah, there is a question in the chat from Gabriel, and I think I should read it for you. Is
Yeah, sure.
asking, during antigen conditioning, how does antigen conditioning influence the generative model's ability to produce diverse yet specific antibody sequences? I think maybe from the previous discussions.
Uh, yeah. This is a great question. So now I'm going to talk about this specific part, that how so because this is a failure mode of the current antibody baselines, they are not able to do that. So that's why we call them, they have like this antigen blindness property. So they don't condition on the antigen properly. And we want to address that like with this like gated thing, that we also add like a bypass path as well, so that we force the model to look at the antigen like every time it's going to make predictions. So like we try to like tackle this, and the current methods, they don't usually do that. And I'm going to talk about now like how in the decoder we are addressing this. Like in the encoder, it's mostly not like much new, but in the decoder, we will apply these ideas of multiple choice learning and try to force antigen conditioning. Yeah, that's our goal here. Yeah, so like and the other part of the model architecture is the decoder here. So in decoder, basically what we do is um, like so what we notice is that the current methods, they use a simple multi-layer perceptron at the output, and then they get the sequence predictions. So like for example, for each position, there's an MLP, and it produces like logits for 20 different amino acids, like, and then most probable one, and then you apply a softmax, and then you compute the cross entropy loss based on that. So that's really like a classic setup. You just have an MLP layer, and then you get the outputs, like whatever we do in classic machine learning, right? What So we identified this problem that because like for antibody CDRs, there can be multiple correct answers. There is no one correct answer. So there's like no one correct amino acid. There is a distribution of amino acids that are plausible, so you can sample from those, and your model should be able to generate it. So in this case, like that's why like the model is usually like resort to the most frequent ones, is because they try to predict the correct one, but there's no one correct answer. There's no one correct amino acid that should be that should the model should output at each position. So what we do here is, so we apply this idea of multiple choice learning. So instead of one like prediction head, the one single MLP, we take like n different like k different heads, prediction heads. So think of it like uh, each head will learn something different about the input. So each head will output some different predictions here. So like head one produces, for example, y, x, g, g something. Prediction head two produces some other sequence. Prediction three produces some other sequence, and then you aggregate their predictions, and then like you keep on doing it for the entire training process. So like this approach is basically like called the winner take all approach. So whichever is closest to the data distribution now, like in later epochs of the training, it gets more weight and the other ones get down weighted. So there's like initially everyone has equal likelihood of producing like its own predictions, not much penalty, but and slowly with training, like you see here that for each epoch we reduce that temperature that we have, so like it reduces the randomness in there, and so it focuses more on the one that is producing more correct predictions. So it kind of relaxes like during the training process. So that's why like it like enables it makes the model learn the patterns, whatever is like more likely, rather than like one single amino acid. So how do we do that? That's like the broad idea here, right? So we implement it like with the idea of mixture density networks. So mixture density networks are usually are a type of like neural network that they produce a distribution rather than one point-wise prediction. So point-wise prediction is like this one correct, one like true prediction, one true answer, and you compute the cross entropy with it. But with mixture density networks, you produce the distribution of like which amino acids are likely, or they're plausible in that position. So it produces like rather than pointwise softmax probability, it produces the probability distribution. So like rather than like yeah. So that's the idea that we implement here, and we also implement. So how does it look like in our specific architecture? So because we have like k prediction heads, so like for example, you see that J1, J2, J3, J4, like they are all producing like these are different matrices that I will talk about in a while. Like they are also learnable. So each one of them produces different logits. Like logits are basically for each like for each position, like for the entire length of the antibody, and then we also produce like these weights that get like that the model learns with as the training goes on. And here is like the other the so what we do is, because each head is producing like its separate logits, we aggregate them together, and then we also learn a coupling matrix. So let me show you how the coupling matrix looks like. What do I mean by that? So now that like we saw that the model produced like for let's say four different predictions, right? And four different logits. So the loss would be the combination of all those four losses. Apart from that, we also, for each prediction head, we also learned that this is like from the classic bioinformatics, like this approach is like, called like the pots like modeling. So pots like modeling is basically that you like there's like preference for neighbors of the amino acid in the sequence. So for example, for position I plus one and position I minus one. So there's like a preference in terms of like which ones can be like immediate neighbors of each amino acid. So it forces the model to not produce predictions that are less likely, that are not plausible in nature. Let's say two amino acids with positive charge can't be next to each other. So this post-like model learns this coupling matrix, like 20 by 20 matrix, and each head is going to learn this separately. So you see like here, like because there's like four different prediction heads, each head is learning this separately on its own. So whatever whichever produces the correct and most plausible predictions, that one like takes the take like that one like will be like taken for the inference stage, and the rest will be discarded. So this is all happening during the training basically. So you see that like we have it's like a belief propagation here. This is also referred to like in computer science. So like the simple idea here is that we want multiple predictions, and we don't want to enforce the model to do that, to predict only one output, but multiple outputs, and then after certain training iterations, you end up with something that is more accurate, that captures the data distribution better. So this is so everything is so apart from the because this is like the model architecture that we develop, so we also want the objective function to do that, to optimize for. So in this case, it would be like the sum of as I mentioned before, sum of the cross entropy loss plus this like pairing loss, or the coupling loss that we have. So it's not plain cross entropy now, but it's also a combination of cross entropy plus pairing loss, and this would be like your sequence loss basically. I think there's a question in the chat, right?
No, it's not a question. It's a note to members.
Okay, yeah, yeah. So now that
Yeah.
we have gone through the architecture, the how like the decoder and encoder both are developed. Like you can also look at the details on our paper further if you want to learn more. Um, so now let's talk about the how we train this model basically. So because we applied like different ideas here. So we have like now like a joint objective that is like also for sequence reconstruction, the one that I just mentioned about cross entropy plus the pairing loss. And then we also have like because we also want to predict the structure. So structure is basically like the three-dimensional structure, the PDB coordinates. So we also have like a smooth a function or a Huber loss, basically. Think of it like simple MSE loss between the ground truth and the predictions. And shadow Pareto loss is so it applies only to the interface amino acids, not the entire antibody, but to the interface amino acids. And then the other the contribution that we did here was that to impose more diversity in the outputs, we apply this idea of generative deterministical point process. So the idea is that we want to match the eigenvalue spectra of the ground truth and the predictions, so that there is they have like if you want to look at your training data, and then you also look at your predictions, so that like there is like enough orthogonality between the predictions that you're producing. So it enforces that it doesn't produce it doesn't help with the accuracy, but it helps with the with calibrating the model better and producing like enforcing to produce more diverse outputs, not like single output that we saw here. So this is like another loss that we implemented as well. And the fifth one is like the antigen conditioning loss. So we add this idea of like cycle consistency that you might know in GANs, is that you want to predict the input as well in the output. So we call it like so we also want to predict the make the model predict the antigen that it got as input. So there's also like this influency loss. This also like in this like directly or explicitly enforces antigen conditioning, because we are penalizing the model if it's ignoring the antigen that is we are giving it as input. So this also like enforces like better antigen conditioning, because like the model is being penalized if it predicts a different antigen. So that's why like it shouldn't be like blind to the input, but it should produce better outputs. And so this is the pseudocode that I've talked about everything, and these are the evaluation metrics that we evaluate based upon like recovery rate, sequence recovery rate. So we have like three like groups of predictions: sequence recovery rate, structural similarity, and then the interface metrics. So like different like evaluation metrics that we evaluate the model upon. And we work with like this camera bench data set that we developed as well. So it's like a new benchmark. You can go and check it out on our GitHub. Like if you want to, if you're interested in antibody design problem, you can go and check it out. This is like a like a recent data set published in iClear GM. So so here we have like three different splits. We have like predefined different splits, and we use these splits, and then we test our model and like train and test to see like we're able to get like improvements in terms of recovery rate. We are able to get improvements in terms of structural accuracy. Like we are getting better structures as output, and also we are getting better interface metrics as well. So they're all like should be interconnected to each other, but we see that there is so the most difficult like the most difficult part in the antibody design problem is the CDRH3, because it's the most diverse, and it defines one it differentiates one antibody from another antibody, and it's really harder to predict. So you see that H1 and H2 are like comparatively easier. Most models do good well on that, but H3 is the most like difficult one. And if you are able to do some good some improvement on H3, that's a significant improvement. And yeah, that's all. So thank you for attention. I think yeah, we are almost on time. Yeah.
Yeah, sure. Thank you very much, Mansoor. This was epic. And yeah, it's interesting always to know that graph neural networks still are within the business of antigen and antibody prediction. For me, that's amazing. But there is a question here from Alfred, and he's asking, in the limitations, you note that contact amino acid recovery remains challenging and may require explicit side chain or physics-based modeling. Have you considered integrating structure refinement or energy-based constraints into AgForce? Could this have any impact?
Yeah, this is a great question. So like um within like an antibody design in general, or with protein design, structure refinement, or like modeling the side chain atoms as well is equally important. And there are like different models to do that. We have not tried it for AgForce. We wanted AgForce, so our focus was more on addressing the specific failure modes that exist in the current methods. And some of the current antibody design methods, they do apply like Rosetta based like structure refinement, or they also like do like sidechain packing like with Rosetta, and also like some energy function that they optimize for. But we have noticed is that like for antibody, specifically for antibody design problem, there's like much more like systematic gaps in our understanding of how should we design these methods. So there it's not like straightforward that like some energy functions would work, like, you optimize for them, and because we don't have enough data to do that, and the data doesn't have ground truth labels for that. So we don't have labels for binding affinity, we don't have labels for like whatever is like more like directly related to bind a good binding partner, or good binding score. We don't usually have like the in the labels for that, so we can't do what we can do like post training, like use RL to optimize for some energy reward. Like some of the baseline methods, they do that. We have not tried it for AgForce, like, but we impose it, like we develop AgForce of with a different focus. We're developing, like we have developed other methods where we work with these energy functions, but to yeah, like we identify, we tackle certain failure mode and try to address that like architecturally, or to produce like some give some novel insights into the architecture design. But yeah, like that's a very interesting direction, and that could be improved more. Some methods still do that. They also do with like RL, reinforcement learning, like post training, like objective, like they have like some energy function that they optimize for. That could be for the different kinds of forces or different kinds of interactions that exist. But yeah, like it is a little bit tricky for antibody design, because yeah, like we don't have enough ground truth labels and enough data to do that. Like to improve further upon that, but there could be like architectural improvements that are possible. Yeah, that's a great question.
Okay. There is also another one from Gabriel. He's asking, what are the most promising therapeutic areas where antigen-conditioned generative antibody design could make an immediate impact? I think it's about applications here.
Yeah, so I believe, yeah, Dr. Patterson could comment on that. Um, but yeah, like because yeah, it's a very it's a recent problem. It has not been like yeah, we have not been able to do much compared to other like biomolecule design, like other therapeutic designs, peptide or enzymes, or like those have like comparatively been like studied better and studied well, and we have better modeling models for two or ligands. We have been able to do that, but I think antibody design problem itself is so yeah, it's going to stay for a while. Like because yeah, we are still like way short, short of like getting developing some open source method that is as accurate as AlphaFold, let's say. But there's some I believe there's some like commercial softwares that are mostly sequence based. There's like some biotech companies that have been working in the antibody design space recently. They have like proprietary software that they have developed, but uh, yeah, but you still need like a lot of wet lab validation to come up with something that is actually clinically useful, or that could have I won't say like would have an immediate impact, but yeah, some groups have been able to produce antibodies that they've validated in the wet lab, and they work. So but like to have like a general purpose and like antigen specific antibody design tool is yeah, it's maybe like not very close yet, and yeah, we have much work to do in terms of modeling. Yeah.
Okay. Professor Peterson, anything for us?
Yeah, I think Mansoor pretty much said everything there. So I think it really depends on how like how we can do some wet lab experiments, like what like we really need to and this depends on like what data we get and sort of who we're so we have a collaborator from the Wistar Institute in Philadelphia who's doing some cancer or no, it's HIV, I think. HIV uh, immunology and yeah, we're hoping we got a couple of data sets from him, and we're hoping to yeah, sort of benchmark our system on that. So that'll certainly give us a bit more knowledge on yeah, how this thing plays out in the real world, right? So
Wow. So I think this means we're still seeing more work in the direction of the possible other applications and within condition generated antibodies.
Yeah, it's very interesting. We started working on this a few years ago, and it's really exploded now. Like there's so much. There's so like it seems everyone jumped on it a few years ago. Like we're seeing it in the you're seeing it in the conferences and all the publications that are coming out. So it's certainly yeah. I mean, I guess like computational drug design came around like uh maybe
But
a
how about
decade
I
before.
need it together?
But,
Even some
yeah,
lectures where I told you to take over, I told you to take over. It was a bit late and you told you to
Alec, I'll mute him.
I'm sorry. Oh yeah, sorry. I thought it was a question. Maybe. Yeah,
No.
yeah. So yeah, like computational drug design came around. Yeah, maybe a decade back, but maybe like nanobody design maybe is something that's like from an applications point of view would be sort of a low hanging fruit, so to speak, because it's sort of simpler and maybe you could actualize that uh, in a lab setting. Because I think yeah, like I think our collaborator was doing that, wasn't he? He was able to synthesize some nanobodies or something like that, right? Was it? Uh,
Wow.
yeah,
Yeah.
and I think collaborators we have another group of collaborators from LUMS in Pakistan. Um, I think they did some wet lab stuff with nanobodies. Yeah,
Yeah, yeah, right, yeah.
yeah. So
Dr. Shoaib, yeah, that's
yeah, exactly right. Yeah.
So nanobodies are a possible application of this work directly.
Yeah, I think they would be easier to do. Yeah, easier to yeah, to sort of actualize in a lab, I guess. So it's yeah.
Interesting. And it is interesting that I've been seeing a lot of research like in the last week about nanobodies. And I think that is a good trigger. I want to thank you, Professor Peterson, and
Thanks very much.
oh,
Yeah, it's
really,
Mansoor, so it's yeah.
thank you very much. And thank you, Mansoor. Thank you very much indeed. Really, this has been very interesting. And to the members, the
thank you for
rest of
having us. Yeah, yeah.
it was an honor
Yeah,
for us. Thank you very much, and thank you to the members. Thank you for attending the session. And I think to, till the next session, probably next week, I want to say goodbye and thank you to Professor Peterson and Mansoor. Thank you very much indeed.
Yeah,
thank you.
yeah. Bye. Yeah, have a good day. Yeah, bye.
Yeah,
All
have a good
right. All right. Bye bye, everyone.
one.
See
Bye
you.
bye.